All right, welcome. I'm Kevin Libuit, joined by Andrew Page. We're from Theogen, and this is the Bioinformatics Lab podcast. And today, we're going to be talking about sort of narrative of tech adoption over the years in pathogen genomics, from software packages to containers, workflow languages, and now at this point of cloud-based GUIs and the different ways in which that's impacting our field. Andrew, you have much more time in the field than I have, so you have maybe a longer time horizon. I say that with absolute respect for your wisdom and experience in the field. But you have a broader perspective of what this has looked like. Because again, whenever I talk about the sort of tech adoption story, I kind of started package managers. But of course, there's beyond that, before. But all this conversation of how do we make this, how do we mature this as a field for interoperability, reproducibility, and all the like. So when you think about the narrative of tech adoption, surely it starts before package managers.
Oh yeah, I remember the dark days where you'd have to hire someone, just to sit there installing software because it would take a week to install one piece of software, because you'd be editing it and trying to find 200 different, dependencies for a compiler that, was last released 5 years ago. So we've come a very long way. Even then, like languages and everything, like I started off in, I guess, Ruby and then Perl, for bioinformatics, and languages change all the time, and no one uses Perl these days, unfortunately. Well, me. But anyway, and then we've all moved to Python. So, things change over time, and we do get better. And actually, genuinely, I think that Perl isn't as good for bioinformatics as Python. After maybe about 10 years, I've come to that conclusion of using it. But then I see like my son is using Rust and he's like, oh, this is amazing, and it's much faster and blah, blah. And it probably is actually, but I'm not going to give up Python, for some new fad language. It's only been around a couple of years. Anyway, so I digress. If you just take the most basic, installing software and package managers, like the big thing a few years ago was just, with Debian, and Ubuntu uses Debian packages. So can you get something in a Debian package, because once it's in there, then you're sorted for life, and Debian Med was the big one for our fields, which is for biomedical resources go in there. And yeah, phenomenally difficult. If you start packaging for Debian Med, they will assign you like a mentor, if someone who can guide you through the process over a few months.
Wow.
Because it's quite, they want these things to work universally and, be working for a long time. So a few months to guide you through the process of building your Debian packages, which would then be sustainable in a long term and kind of easy to maintain. And God, so that was probably first got into it in about 2015. Because I just happened to work with a guy who's one of these maintainers. And that the advantage of that is when you go to command line, you can just have apt-get install blah, like Rory or something like that. And that lowers the bar so much that it makes it trivial. And if you make your software easy to install, then of course people will use it. And that's a key magic trick, to getting people to use your software, is to make it trivial to install. And then, you go one step further, and then we had Homebrew. Do you remember that?
Yeah, I do. That's about when I entered the chat. It's around the Homebrew space.
Which is a little bit, another system. They made it a bit easier to go and install software, but it kind of fell off the rails. It became unwieldy. Conda came along, BioConda, and that's been kind of the default now for people to use, because it just works, and everyone is kind of gathered around that. It's quite easy to install stuff. Dependencies are reasonably okay at the moment. You can get into obviously a bit of a nightmare sometimes. And then Mamba has really helped the installation process, rather than waiting an hour to install one piece of software because things got so complex with resolving the dependencies, it now works. It's pretty quick. So, we've come a long way. We've also come a long way with workflow managers. What was the first workflow manager you used?
Make. I think I would consider Make the first kind of workflow manager. I was writing Make files where I was like writing shell scripts of, just the executables, and then I would compile them to not compile them, I guess, but I would kind of curate the workflow through a makefile, and then I would define endpoints at the beginning, and then it would be, that's how the workflow worked. It was just recipe style. It would define the endpoint, define the process. It's very much the same components. I mean, and then that's where I started learning about snake make, which was really make in Python. And then where I really started running in workflow managers was use of Nextflow. And I think it was actually Aaron Young, again, on the staff B side, who kind of introduced us to workflow managers, and specifically, maybe it was Kelsey Florick, actually. I don't remember exactly, but they introduced us to the concepts of workflow managers. So we kind of went from the conversation of how do I get a single tool to work properly on my machine, ensure that the dependencies are consistent throughout the different environments, but then workflow managers changed it in that we were able to string them all together in a really cohesive way. Sorry, 1 second. But that became kind of a really pivotal point. Again, I'm speaking on the staff B side, but this is something that I think is echoed throughout the field of it's so much more standardized in how we're doing these analysis. You don't have to write your own Python logic to string data from, your aligner to your variant caller to whatever it is downstream for characterization. Rather, there was a consistent language there. Somebody outside of that, of your laboratory, can look at it and understand, okay, this is a Nextflow workflow. I understand the architecture of this entire repository. And moreover than that, I can take the modules from that and build them into my workflow in a pretty seamless way. So that was a huge jump in terms of interoperability, collaborative development that we saw in Staff B. And we never really looked back from that. That became just the status quo.
Yeah, the first one I used was called vertebrate resequencing codebase. It didn't even have a name. Like VR Codebase was the name, and it was built for the Human Genome Project, not Human Genome, the 1000 Human Genome Project in the Sanger Institute. So this was before people were doing things at scale in genomics. So, you're talking more than 10 years ago. And so a lot of, Nextflow didn't exist. A lot of these other things did not exist.
Yeah.
And so they had to be built, and the Sanger Institute was at that time, would be producing maybe 20-30% of the world's sequencing from one place. So they had a scale that other people didn't have, and so that's what we adopted. And we were then adopting it for pathogens, which is quite different to human, because you have to do a lot of things. You've got a lot of small little things, and that's very different to human, which is you've got a few big things that you want to do things on. So it's it's a flipped problem, and of course that breaks everything. And yeah, so anyway, that was the first one I ever worked on, and highly complex. Changing anything was very difficult. It was all in code. It was on GitHub and whatever, but it was all in code, and it was all in Perl as well. And everything was monitored by, say, writing little files to disk, and checking them and whatever. So it worked, but the growing pains came in, and so then the next generation came out, which again was super lovely, beautiful Perl. But again, it was more cloud computing focused, and it did things very well. But then I had seen Galaxy a few times. I was like, oh, that's actually pretty cool. So when I went to the Quantum Institute, that was the first thing I brought in was Galaxy, because you got a web server that anyone can use. It can run on a cluster or on cloud. It's got all the tools, kind of linked in and built in, because people, again, wrap up tools, simple XML files, wrap the tool up and make it work, and can run in with Conda and whatever. And then that allowed a lot of people to do complex workflows. A lot of lab people who don't know the command line, moving them away from this kind of black screen and a blinking cursor, and they don't know what to do. It's to, oh yeah, I can just click here, I can search for aligner, and then suddenly you can align your data, and everything flows in from the sequencers, and then it can self-service to do their thing. And I really do think GUIs are, they're worth their weight in gold. You can see the amount of stuff that people can do, say even in Excel. You got power users of Excel can do phenomenal things if you just give them something straightforward, easy to use, good usability. They can work wonders. When it comes to the command line, obviously that's very powerful for doing things at scale and speed, and for joining things together. but you have a very high barrier to entry there for an ordinary person. Even someone who works in bioinformatics, it can be a high barrier to entry, because you might have to read through lots of documentation to figure out how exactly does this fit in, what exact format does this come out in. You shouldn't have to solve that over and over again. It's nice when you can just kind of have them linked together. Someone solves it once, maybe defines the outputs, say for Nextflow or for Galaxy, and then it's there and it's forever more works. Yeah. Anyway, workflow managers are fantastic, as are things like solving dependencies with Conda. I just love that. I mean, Jesus Christ, like the pain, the absolute pain you used to have to go through to install software. And if you use clusters like the kind of old school HPC, like basic physics clusters from back in the day, massive, machines in a big room with blinking lights. Actually, installing stuff in those is a right pain in the arse, right? Because you have one base operating system, and you kind of install stuff and load in stuff, but like the operating system we had, that particular, whatever was installed probably 5 years ago, that's the version you're stuck with. That's the version of compilers you're stuck with, and you can't change anything. So actually having the ability to run Docker and Singularity and whatever is amazing. And that, I remember the arguments people had, about allowing Docker to run on a cluster. Like, no, that's security risk. We can't be having that. Singularity did help out a little bit with that, but it's we've come a long way, and now we're in the cloud. Like, I mean, Jesus, that's amazing. You can just spin something up in a few minutes. You don't have to wait 2 years to go through a procurement process and buy stuff. You can just bang, there you go, have the resources, run stuff, and you have full control over it. Then you can tear it down.
And that's often the kind of three, in terms of tech adoption, those are like the three turning points I always have in my mind. First is sort of, maybe I'd put a slash in package managers and containers, where it was solving the problem of dependencies and install and portability of the single tools in itself. And that's solving a lot of the problems that you described. And then, so now we can all, if I have a tool, if I have an assembler, I can run it in a really reproducible way in my environment, and you can run it in the same way in your environment. Then the sort of next big tech adoption was the workflow managers. Not only do I have the same tools, I can string them in a way that makes it plug and play. And as you mentioned too, these workflow managers not only help to standardize that, but they also standardize the running of them. I can run it on an HPC, I can run it on a local VM, or I can run it in the cloud. And all the workflow managers now have built-in capabilities for that scalability. And so that, you briefly mentioned it, is that third layer is the GUI, the web applications that kind of wrap all these things together. And my first experience with that was using Galaxy, actually through Genome Tracker. They adopted Galaxy as a platform, and I realized, oh my goodness, I don't have to teach CLI any longer. Rather, I could teach them to click a couple buttons, I can show them the workflows, and I can focus on the results and the analysis and interpretation, rather than your Linux environment, your library directory structure. This CD means you're changing folders or something like that. And I think I'm stealing this from, I think it was Peter Van Houston in Cape Town, where he said a line, and I don't think he meant to be codify this, but I thought it was a really powerful line, that in public health they should be doing less bioinformatics and more public health. And I thought, oh, that's kind of a nice little line. It's like, yeah, we should really be working to abstract the sort of technical nuances of bioinformatics, and really instead provide them with tools that allow them to do public health. And I think GUIs, the graphic user interfaces behind that, is really what allows us to kind of traverse that problem, where you don't have to be card-carrying bioinformatician to do bioinformatics. Rather, you can use bioinformatics as a tool to inform your public health that you're implementing there. And obviously we've seen that. I mentioned Galaxy. Terra has been an incredible resource where you've seen that kind of come to play. And then you see, again, I'm stealing these terms from different people. Ali Black, in her 10 recommendations for pathogen genomics, she coined this term of an open bioinformatics ecosystem that's built off all these technologies we've talked about. Containerized algorithms that are written in these standardized workflows that are made accessible through these GUI portals. And it's like, oh, okay, once you find that mix, we've seen that that model allow us to distribute bioinformatics tools to laboratories across the world. We've seen that happen in so many different ways. We recently put out a publication where Terra from the Broad Institute has fit that model of the open bioinformatics ecosystem, in that it's got a really well-maintained graphic user interface, cloud backend with GCP. It's got the doc store repository, which is, you know, coming out of the GA4GH universe there. And then it's because it's standardized workflows, you can also have the standardized outputs, where that you can then transfer these outputs into different systems, be it transfer it to, you know, SRA and NCBI for distribution and international accessibility. You can also transfer these results into maybe more secure environments, where you might be combining things with sensitive metadata for genomic epi investigation. So watching the tech adoption happen, it's gotten to us a point where now it's just these resources that we've all been toiling with and trying to make sure that work on a machine now are in the hands of laboratorians, so that they can use these things, generate the results, and make sense of the data in real time, inform what's happening on the, you know, either infectious disease side, be it public health, clinical, food safety, or otherwise.
And you can see what happens, you know, when things go wrong, when things are not open, like Biomerics. The company has decided, okay, there's not enough money to be made to maintain this, you know, for the public health world, so we're just going to shut it down. And that's it. And that's a closed source, locked away, highly critical tool for public health, and it's going. And had that been open, it would have been very different, because then obviously, you know, the community could take it up and, you know, keep maintaining it and extend it and whatnot and keep it alive. But if a commercial company holds the rights to that, you know, the source code and how it works and all the infrastructure behind it, then it's a problem. And so we really do need to have open bioinformatics, open tools, open databases, open everything.
Absolutely, and that's definitely been part of the ethos in, you know, how we work professionally with public health labs, is that we feel as if it's funded and supported by public health and it's applicable to a single public health lab, it's very likely going to be something that other public health laboratories can also find utility in. You know, for example, we work with laboratories in Mozambique, helping develop, you know, assays for HIV sequencing and analysis. These same resources that we're working with APHL Global Health to develop and innovate upon in Mozambique, we're watching being proliferated to laboratories in the US who have the same interest. And again, getting back to that ethos of, if we're going to develop this tool publicly funded, it needs to be open source and open accessible, because it doesn't need to be closed off. That's not necessarily the business model that's conducive to ongoing support long-term of public health laboratory pathogen genomics here.
Absolutely. On the tech adoption front, actually,
Yeah.
I was at a conference there the other day. What was it? Science Meets Policy, using next generation sequencing to tackle foodborne threats, at the European Food Standards Authority. And that was
Yeah.
really, really eye-opening, because all the different, you know, each country in Europe, you know, taking a slightly different way of doing things. But, you know, obviously everyone is doing public health and everyone is doing food safety, and you want the same end goals, more or less. And it's very interesting to see how people are approaching different problems within the context of their own country. Some, you know, are going very much on the I suppose, short read, you know, bang stuff out, do CGMLST, share the CGMLST results. Others, you know, are very much more, well, you know, we'll release the data or we won't release the data, or whatnot. So yeah, very interesting to see how people are approaching the same problem.
Yeah, tech adoption, it's big. Yeah, we've only really talked about it on the sort of dry lab side of things, but also on the wet lab side of things. How are people adopting different sequencers, different platforms, from Illumina to ONT? I think you have some perspective on ion torrent adoption and things like this too.
So yeah, I was a bit horrified to see some benchmark sets, and they had like ONT, sorry, they had ion torrent data, but not ONT data. And I was like, come on, like, you know,
Yeah.
ion torrent, the last time I saw one was actually is wrapped in plastic waiting for disposal in an underground car park.
That's
You
that's
know?
right. It's the first sequencing data I ever generated was actually on an ion torrent PGM. If you're familiar with that. Yeah, yeah, we
They
have that
were
in my
they
lab.
they had the little kind of Xbox logos or something on them as well,
Yeah,
didn't they?
yeah. They even had, I think, a slot for your iPhone if you wanted to, you know, put some tunes on while you're preparing those libraries.
It was an iPod or something silly like that. It's a really old connector as well. Yeah.
Yes, yeah, 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 yeah. It was
Oh.
that was my first time generating sequencing data. We had those ion torrent chips. Uh, I'm diverting there, but that was my first tech adoption into it, was ion torrent, and it was interesting because, you know, again, not to, I don't think we're saying anything that's wildly controversial, but, you know, knowing about the data. But every time I would put the data out there, people would talk about the the error profile kind of associated with what was being generated there. So it is interesting watching benchmarks getting generated where you have a single technology with a known error profile without maybe also adding some context with either Illumina data or ONT data, especially for benchmarks.
Yeah, and what was very eye-opening was some countries don't allow people to use the cloud for security reasons. So it very much limits them on what they can do in terms of mathematics and in terms of scaling up as well. They can't just go, we need more, bang, there you go.
That's a huge point in talking about tech adoption is cloud computing. That because that was a big conversation is infrastructure development across the US that we were always a part of in staff B, and people were doing pretty much everything you could see in terms of on-prem servers, HPCs, cloud, working with academic HPCs and all the like. But then it just became so obvious that cloud was the solution for all the reasons
Yeah.
it is in every other industry. So that's been a big thing, but it took a while. Like even at me in Virginia, it was like 2.5, 3 years of discussions with our IT before we were given our AWS account. But I think we're at a critical mass where we've seen so many laboratories kind of break the barrier, have the conversations with our IT, that those conversations are shortening dramatically, and you're seeing wide adoption across really the world now.
I know in academia, like when I came into the quadrum, it was all traditional HPC, big cluster in a server room on-prem. And we moved over to OpenStack, which is a private cloud, which is a million times better and more flexible. So, that's where we started down. And I'm sure in the future, it'll become public cloud, because it's very easy to go from an OpenStack private cloud to a public cloud. But what during the pandemic is fantastic was that all the COVID sequencing was being done and uploaded to an academic cloud called Climb, MRC Climb, which is OpenStack based in 3 different universities in the UK. And so instantly resources were available, like no one had to sit around and say, oh, can we rent some resources from here or borrow some from here? It was just like, bang, there you go, all available. And then the public health authorities, like UKHSA, were using Azure. And everyone was just cloud, either private or public. And there's no sitting around, waiting to buy vast amounts of storage. It was just like, we'll use what we can right now and solve these problems. I know other countries struggle because there's legislation that says, I don't know, they can only, if they were to use the cloud, it can only be within their country or within one particular region. So it kind of makes things a bit more difficult. Obviously, Amazon and Google and Azure, so Microsoft are doing a good job by having data centers all around the world, but it is a problem. And then when you get into clinical stuff, a lot of stuff can't even leave like the hospital, some really sensitive data, you just can't cross that barrier. When I worked at Quadrum, the building we shared with the local hospital, and obviously researching hospital, we had 2 separate physical networks in one building, in one kind of server room as well, 2 separate physical networks, because they had to keep, the clinical stuff totally separate from the research stuff. Research is seen as, obviously more loosey-goosey and high risk than the clinical stuff, which is fair enough. But anyway, hopefully with time, people will adopt the cloud a bit more.
Yeah. And before we end, the last or I guess we talked about with the historical tech adoption. Now looking forward, I think, you and I have had a couple episodes on this, but what we're seeing is the big tech adoption that's really going to be changing the field is, machine learning and AI and how it's going to impact things. And we saw little murmurs of it, even like with Pangolin. There was the Pangolin Learn, and it was like, okay, how are people going to deal with this sort of black box of decision making? And it wasn't taken too kindly. I mean, it was taken in its utility and practicality and speed, but it was definitely a huge preference to the phylogenetic placement, because people could make sense of the Usher approach versus machine learning. But it's going to be interesting over the next couple of years where more of this adoption of machine learning and AI in general there, or more specifically, I guess, is going to be interesting to see how it impacts our field. I don't know if you have a hot take in the last couple of minutes before we close out in the tech adoption of AI and in pathogen genomics.
Honestly think it's going to change things in ways we can't even comprehend right now. It'll just be there, it'll always be on, it'll just be part of what we do. And I'm excited for the future. I'm excited to do less work. I love now when I program that it saves me so much time having to type stuff out, because like it can pick up all the obvious stuff that I'm probably going to do.
Yeah, because we're already using it in active writing, active programming. I'm really interested to see how the big tools kind of come in here, because a lot of what we do is a lot of classic categorization that seems well for the taking for these kinds of technologies. But I think it's going to be the policy and adoption of how do we, to what level of trust we put to this, in knowing that we can't really necessarily just open up and understand how we came to these decisions. So that's what keeps our jobs exciting.
Absolutely.
All right, good episode. We'll continue on this conversation, I'm sure, in future ones in the coming weeks.